Bismillahirrahmanirrahim. This is Mahnoor presenting a paper based on a framework for white blood cell segmentation in microscopic blood images using digital image processing. The main objective of this paper is to segment the white blood cells into its two dominant elements that are nucleus and cytoplasm. 20 microscopic blood images were tested, and 92% accuracy for nucleus segmentation has been acquired and 78% for the cytoplasm segmentation. The results indicate that the proposed framework is able to extract the nucleus and cytoplasm region in a white blood cell sample. The white blood cells or leukocytes play a significant role in diagnosis of different diseases, whereas a few complications in extracting the data from white blood cells occur due to wide variation of cells in shape, size, age, and position. So the goal of this study is to segment the white blood cells, nucleuses, and cytoplasm using this framework. A hematologist can play a vital role in automatically processing blood slides for the first screening in detecting diseases. Through using techniques, they can find the cell counts and the ratios of nucleus versus cytoplasm to identify and classify different types of white blood cells, such as neutrophils, basophils, and lymphocytes. So, in this paper, a framework has been proposed that consists of several methods that integrate together for nucleus segmentation and cytoplasm extraction. And there are a lot of segmentation methods. Some common segmentation methods are edge and border detection, region growing, filtering, mathematical morphology, watershed clustering. And a lot of people have used these methods in their studies. Is like Ritter et al. Had used fully automatic method for segmentation and border identification. Ongen had used morphological pre-processing. Snake-Mahnoor algorithm. Jiang has proposed white blood cell segmentation scheme on color space images using feature space clustering techniques, scale space filtering for nucleus extraction, and watershed clustering for cytoplasm extraction. Similarly, Lazia et al. Had used morphological operators, and Scotty had presented the automatic morphological method. And finally, Kumar had used a Tiga energy operator for segmentation. So the proposed framework for this study is to separate white blood cell segmentation or the leukocytes from the other components of the blood. And the typical blood smear image consists of four components, which are background, red blood cells that are unnucleated, white blood cells that are nucleated, and the cytoplasm. So the white blood cells appear darker than the background, red blood cells appear in an intermediate intensity level, and the shape variation in cells is in their nucleus. So the proposed framework of the segmentation scheme is basically they have got the color scale images of the microscopic blood samples. Then they have converted those color images into the grayscale, and then for getting the sub-image separation, they have got the sub-image separation of white blood cells, and they have applied edge detection algorithms. After that, they have applied gradient vector flow algorithms for finding the nuclei using the snakes, and then they have filled the holes. And once they have found out the nucleus, they have segmented them. Once segmented nuclei, they have subtracted the nuclei section from the grayscale images, and they have applied Zek algorithm for finding out where the cytoplasm is. And once found, they have segmented the cytoplasm as well. Nuclei have variable shapes and different kinds of leukocytes, and it is quite challenging for segmentation of nucleus and shape modeling. So there are different methods for segmentation, among which the active control models have gained quite attention. So the snakes are deformable. Snakes are also known as the active control models. So snakes are deformable curves, can move and change their shapes to deform boundaries of the objects in an image. Curves are defined within an image domain and which can move under the influence of forces, maybe internal or external. So the internal forces are within the curve itself, and the external forces are derived from the image data. The forces are defined in such a way that the snake conforms on the object of interest or the object of interest and its boundaries or the other desired features within an image. There are two types of active control models. One is parametric and the other is geometric active controls. The geometric active controls or models or geodesic snakes have been proposed to address the fact that parametric active controls models cannot resolve topological changes. For this pre-processing scheme, segmentation is done on sub-images, so there are no topological changes since only one object of interest exists in each sub-image. So in parametric snakes model, a traditional snake curve is defined, which is defined as SS of x, which moves in the spatial domain to minimize the energy function. So alpha and beta are the weighing parameters, ESC as the external energy function, and S of x, S of s, and x dash of s of x, and s double dash s of x are the first, second derivatives of the function. Image gradients can be used as internal or external forces basically in the parametric active controls. So the gradient vector flow, due to its insensitivity to initial positions in larger capture region, is considered as better gradient based model, and it points towards the object boundaries, and which is when addressed to closely to the boundaries, and it works very smoothly over homogeneous images. Some more equations are applied for the image segmentation and identification of nucleus and cytoplasm. There is one method that is known as scanning edge detection. This edge detection method do not misses the edge occurring in the image, and it would be completely non-responsive towards the non-edge areas. The steps are, first of all, smoothing by using the Gaussian filter to reduce the noise and unwanted details. There are some equations for this. Gradient calculation also occurs by using the gradient operators. There are equations given for that, and similarly for threshold, finally, there is a piecewise function. So the next steps are, and this process is known as no maximal suppression. The next steps are rigid pixel thresholds, which work on T1 and T2 with T1 less than T2. So the rigid pixels with values between T1 and T2 are weak edge pixels, and those with the values larger than T2 are so strong edge pixels. Edge segments in T2 are linked to form continuous edges. To do so, each segment in T2 is traced to find its end, and its neighbors in T1 are searched to find any edge segment in T1 which can bridge the gap until reaching another edge segment in T2, which is actually the whole film. And by the edge detection method, central connected object boundaries that represent the nucleus are clearly obtained. In next step, GVF of the images were calculated, used as internal and external forces to guide snakes to deform nucleus boundary edges. Nucleus is the connected boundary in image and has been filled up by following instruction to have a clear segmented nucleus. Following are the few functions and equations which are applied to the images. Finally, the results are 20 blood images from the acute leukemia cases type L2 were captured using microscopic Olympus BX51. The system was a soft imaging system with analysis software. Camera used as CC12. Magnification is this. 400 for pictures, and amount of fields per slide acquired is quite random, 2 to 3 fields per slide, and total size are 10. So the results are for nucleus and for cytoplasm. So for nucleus, these are the original WBC subimages, and then those are converted into grayscale images. Then the edge detection algorithm, which is Kenny edge detection, is applied to find out the edges. After that, GVF snake algorithm is applied to find out the boundaries of the nucleus. Once that has been identified, the nucleus has been extracted, and finally the results are shown in the last image. And for cytoplasm, this is the image after subtraction of the nucleus from the grayscale images, and then this image is actually the detection of the cytoplasm, and then they have filled the segmented cytoplasm. And now, on average, there is 92% accuracy which they have got for the nucleus segmentation, and for cytoplasm extraction, they have got 70% accuracy. Since cytoplasm segmentation process depends on the result of the nucleus segmentation, so there is 8% error on whole. So on average, they have also found that there are 55%, there are 55 WBCs in a typical blood smear image. So there are chances that some parts of the cytoplasm and the nucleus to be missed in each WBC. The results show significant accuracy to be used for further analysis of blood images on detection of acute lymphoblastic leukemia or any other disease related to white blood cells. The conclusions are actually in this paper. They have demonstrated a proposed framework for segmentation of white blood cells into its constituent parts, which are actually nucleus and cytoplasm. So for nucleus segmentation, they have got 92% accuracy, which is based on the morphological analysis, and for cytoplasm extraction or cytoplasm segmentation, and they have got 78% accuracy, which is based on a pixel intensity thresholding. That's all.